So we're going to pick up right where we left off in class, talking about, so our previous slide was talking about neuronal death, and we were starting the process of talking about specifically how it occurs in neurons. Now, I would not expect you to know very detailed process, but I do want you to know just generally how this works. So our cells have death genes. These are expressed only during the process of apoptosis. And the way that it can happen is if, for example, a particular cell had some damage to it from DNA, some particular DNA damage, and it just couldn't survive. A virus can cause these death genes to become expressed, as well as lack of nutrients to the cell. And we're going to talk about that here in a minute. But really, the key part here is that if you look at the mitochondria, when if a cell has a particular influx of calcium, right, that sort of starts the process of apoptosis. And so we have a couple of really important players here. So we have caspases, which is a family of proteases, whose job it is to basically cut up proteins and cut up DNA and destroy the cell. Now, your cell has these, but what sort of stops it from activating are these inhibitors of apoptosis proteins, or IAPs. So IAPs' job is to inhibit caspases from cutting up proteins. And so if you block IAPs, then that will then cause caspases to be able to function. So what happens is this in the mitochondria, we get this influx of calcium, and then apoptosis is beginning, and so it releases a protein. The mitochondria release a protein called Diablo. And then Diablo is going to bind to the IAPs, and it's going to inhibit them, which means that they can't do their job to inhibit the caspases. So without that inhibition, the caspases then take over and cut up the proteins in DNA. So essentially, if you inhibit an inhibitor, it's going to cause a reaction. And so in this case, it will then dismantle the cell. Now, there is a protein, BCL2, which can block apoptosis from happening by preventing the release of Diablo, therefore allowing the IAPs to do their job to inhibit apoptosis by inhibiting the caspases from dismantling the cell. But let's talk about one of those factors and how it can actually lead to neuronal death. One of those is sort of lack of nutrients, lack of what we refer to as neurotrophic factors, which are chemicals that are necessary for the survival of the neuron. In two of these examples, we have the nerve growth factor, NGF, and then we have the brain-deprived neurotrophic factor, which is BDNF. And so the nerve growth factor is important for keeping the cell alive, but also it is it's produced by these targets, which we'll get to in just a minute, and the axons of these innervating cells are the ones who are sort of taking up the nutrients. And then we have the brain-derived neurotrophic factor, which does something similar, and is providing the neuron energy. So I like to think of this as a bit like the game Hungry Hungry Hippo. This is just kind of the one thing that kind of always pops in my head. So we have these target cells, and they're releasing these neurotrophic factors. So think of these as like the little balls, right? And then this is like the little hippo coming out, and it's trying to gobble up as much of this as they can. And so this is the axons sort of taking in these neurotrophic factors, and then they're transporting it into the soma or cell body. And so if you have this particular one sort of taking in, it's innervating, it's active, it's sort of doing its thing, and it's gobbling up all these neurotrophic factors. What happens is that this particular cell isn't going to take in enough to take it to the soma to be able to survive. Same situation here. We have one that's not getting enough in, and this one is getting more in, and it's essentially these cells here are going to then start that sort of catalyst process of apoptosis, because they are not going to be able to survive not having nutrients. And so this process that happens kind of at the beginning, where we see over time with these cells not getting enough nutrients, they're going to then die off. And so this particular cell isn't able to sustain itself anymore. It has not gathered enough trophic factors, and then you'll see these particular cells die off. Now, this is of course occurring early in development, where we see neurons, sort of competing and trying to flourish. But this process happens sort of early in development. But as we continue to develop, of course, then we see instead of looking at sort of cell life, we're looking at synapses and connections. And so here we see later in development, these axonal processes are now starting to compete. So now we have a more sophisticated cell here, but we still have these target cells releasing these neurotrophic factors. So this particular cell is getting nutrients in both of these synapses. But in this case here, this synapse is getting plenty in and able to take it to the soma. But this particular synapse is not getting enough in. So remember the hungry hippo, this is eating it up, this is eating them up, this is eating them up. But this one doesn't seem to be having as much success getting these particular neurotrophic factors. And it's likely because this particular synapse is not innervating enough. There's not enough activity there. It's just not firing enough to be able to take in these neurotrophic factors. And so what ends up happening is that this actually will die off, and that's where you start to get some pruning, and we see some selectivity in terms of synapses. And then, of course, as we continue on with this process, we actually see synapses rearranging because some of them are receiving more nutrients. We're seeing that some of them are firing, and they're able to establish greater connection across other synapses. And we see an abundance of synapses happening and synaptogenesis happening at different portions of development. And so if you look here, so this is a really nice graph here. The red is the visual cortex, the blue is the auditory cortex, and the green is the prefrontal cortex. So you can see within the first year of life, there's a lot of synaptogenesis. There's a lot of synapses in the brain. And you can see that the visual cortex here is especially prevalent in the first year of life. And we know that experience is very, very important to maintain these synapses. Because if you, for example, don't allow yourself to be in an enriched environment, or you don't have exposure to, if you don't expose an infant to much stimulation, then those synapses aren't going to fire, and essentially they're going to die off. So, enriching environments are really important for the first year of life. And we know that there is kind of an overabundance of synapses during this time, because the idea is that whatever doesn't get used is going to then be pruned off. Think of it like, you have a bush, right? And the bush is just kind of wild. And then you start to literally prune off. And then now you have a much nicer bush here. It's just the same idea in the brain. You have an overabundance of synapses, and then that processes start to actually thin out. And so we see this happening. As you can see here, there's a bit of a dip here, and you're going to see another really big pruning event happen right after adolescence, or right around adolescence. You see another dip in the synaptic arrangement because they're pruned off. And so when we look at the developmental course of the brain, the thinning process starts in sort of caudal to rostral direction. So this is like the back of the brain, this would be like the cerebellum. So we actually see this pruning process happen, whereas this is the frontal portion of the brain. The frontal cortex is the last place to develop in the brain. And so typically, many people think that the brain stops developing, maybe in childhood or early adolescence, but really the brain isn't finished maturing until the early 20s. And so, if we're looking at, as between 21 to 25 years old, because this pruning process occurs. So this pruning process really starts around adolescence, and it doesn't really stop until it hits the frontal cortex, which usually is around, early 20s. So the brain is still actively developing and changing up until early 20s. We also see myelogenesis happening as well, and so those astrocytes and oligodendrocytes, those are going to really begin after neurogenesis is mostly complete, and this process will continue throughout your lifespan. And you know that, and we've talked about this before, oligodendrocytes are what form the myelin in the CNS or central nervous system, whereas Schwann cells are what forms the myelin in the peripheral nervous system. And so we like to think of myelination as a sort of a rough index of cerebral maturation. And we know that, this process is important for, the myelination is important for having really good signal insulation to the axons to make sure that we can have really, really solid action potentials. And so myelination starts late in gestation, continues well into childhood and adolescence. And you can see a really nice chart here to talk about how this myelination occurs. From here we can see birth all the way to 20 years old. And during this process here you can see where synaptogenesis and the synaptic pruning points are, and where they occur, for example, here in the prefrontal cortex, to give you an idea of where that occurs and when it occurs. Now myelination, just as we kind of talked about this kind of synaptic pruning, it continues to happen throughout the course of development, and here we can kind of see how it develops last in these prefrontal regions, because here a blue would be a high level of myelogenesis or myelination occurring. Here you can see this is a really dark blue. At 5 years old, there's not a lot of myelination in the frontal and prefrontal cortex. And here you can see by 12, still not a whole lot of myelination in the frontal cortex. You can start to see it happening about 16, about adolescence, and here at 20 years you can see it really develop. And really where we're starting to see those areas is the sensory and motor cortex here, cortices here. So here you can see the sensory cortex, and it's starting to develop and expand out. And of course here this prefrontal cortex is the last region to really see the myelination sort of finish developing. And here's a really nice kind of scope of what's happening at what point. And so here's conception to birth. And this is sort of directly right after birth here, the first maybe 2 weeks of life. Here you can see that neurogenesis is occurring here. We talk about 7 weeks. And then we can see that process of neurogenesis mostly occurring in this time frame, and then sort of tapering off until right about birth. And then here you can see that during this process, of course, we have migration and differentiation, and then we see these neurons maturing into more specialized cells, into what they are going to be. And then, of course, you can see a really nice graph of the body weight and brain weight correlation to that as in this case, later on, the fetus is developing. Now, we know that especially once a baby is born, that we need to have an enriching environment, because the cells do respond to enrichment. And so if you have a cognitively stimulating environment, that's going to maximize connections and allow for additional synapses to form and all those sorts of things. So it's a really important process. Here you can even see, so if we have rats in two different kinds of environments, we have one that's housed in a laboratory and one that's in an enriching complex environment, you can actually see that there are more connections here, that it's a wider range in terms of where it can connect it with additional arborization and branching that occurs. And so we see that an enriched environment leads to things like larger, more defined and more synapses. And we also see larger and more specifically more astrocytes. And so this is really important for this sort of experience and brain development link. Now, we also see that neuroconnectivity can be affected both prenatally and postnatally. Prenatally, the argument is more of the chemoaffinity hypothesis, where we're seeing, and we saw some of this in the videos that I've shown, that these neurons and axons and dendrites are drawn toward specific chemical signaling, and we know that these particular pathways are guided probably in a sense that is to allow for typical development. If we see incorrect layering, of course, we can see disorders occurring, and we'll talk more about that in a little bit. But these are kinds of things that you can actually see happen in vitro. You can actually pull these independent cells and actually see them, differentiating into specific cells based off of not necessarily environmental influence, but likely because of a chemical influence. And then, of course, postnatally, you're seeing that these connections are being fine-tuned because of the experience and because of the exposure. And if you receive an injury to the brain, especially after birth and postnatally, you're going to see that the brain will try to reestablish those original connections if there is a particular injury, if there's something occurring. So if there is something that happens after these connections have been established, then we're going to see those connections to try to continue along the original pathway. Now, we have determined that there are probably a few very critical periods of development. We've kind of identified critical periods of sort of sensory development, language development, things like that. One particular window of development that we've seen some really interesting studies on is, for example, vision. So this is an example of 3 different experiments that were done. This is a sort of historical set of experiments done on cats. Here you can see that we have sort of a control group where we have no deprivation, the cat's eyes open, and just to kind of see what a typical neuronal map looks like, right? So, during infancy, during adolescence, and adulthood, and this is a typical developing cat, where we see, the basic neuron structures that have developed, and then over adolescence we see arborations, and we see these, a stronger connectivity pattern, than all the way to adulthood, where we see a much more mature series of neurons. And then if we have a kitten whose eye is sewn shut very early on, and here you can see exactly when in development this particular kitten's eye was sewn shut, you're going to see an abnormality in that particular kitten's eyes. And so if you compare, so if just the right eye is sewn shut, so the left eye gets to develop normally, but the right eye is deprived of visual input. You see here that these neurons in adulthood look and in adolescence look very much like the very early neurons from sort of before they were covered and before their eyes even opened. And so what ends up happening is that because these neuronal projections are not forming because they don't have the exposure, they don't have visual input, then if you uncover this eye, then what's going to happen is that that kitten will be blind in that eye because these projections are not matured. They haven't developed properly, and so we've identified that this must be a window, because if you repeat this experiment but do it later in development, here you can see that this particular cat's been able to actually develop a proper, in this case, right eye series of neuronal connections. Now you can see that if you open this eye and test the eye, you're going to see that those connections are still established. It's not going to blind the cat. Now, some may question, okay, if we come back over here and the cat is blind and not able to see anything, we do find that some in cases where we see this happening from birth, we can actually see some connections reestablish as long as the eye gets uncovered and now exposed to visual input again. Now, does that mean that the eye is going to be as fully functioned as it was if it was never uncovered and never covered? No, we're likely going to see that this process is going to take time, and the cat is going to regain some vision, but it may not be to what it was. But we know that it's especially detrimental if you do not allow for particular input during this very critical window. So if you allow these connections to establish after birth, here you can see at 12 weeks, if the cat's eye is then sewn shut, then you will not see the same kind of neuronal issues as you see if it were covered at birth. And so that does give us, some insight on what happens if an infant is injured in utero or right after birth, and what happens in terms of these connections if input isn't allowed into a particular area of the brain. And so we know that there are some areas that are very critical to that development. So one thing that we know, that early deprivation of sensory experiences. So if you place an infant in the dark, for example, it is going to, you're going to see atrophy of those dendrites. Versus if you take an infant and put them in a very cognitively stimulating environment, you're going to see growth of those dendrites. They're going to be forming typically, and they're going to expand depending on, and you'll see more dendrites, you'll see more connectivity, you're going to see more dendritic spines in a stimulating environment. Now, if you deprive an animal or a human of early social experiences, so for example, there have been series of spider monkey studies where they actually took these are the Harlow studies, they took the monkey from the mother, and they wanted to see very early on to see if that would impact the social behaviors later on in life. And we do find that it absolutely does have a profound negative effect on later intellectual and social behaviors. Essentially, the animal doesn't know how to act. We also see this occurring in very, very severe neglect cases in human children. So if a child is sort of kept into a room with very little exposure to very little social interaction, we'll see that child typically will have some serious intellectual disabilities as well as social behaviors, and may even sort of not necessarily lose completely, but really lose the typical ability to be able to use language and understand language. So if we see a human where deprivation is relatively short, we can see some recovery, but if this is several years, then we see, of course, some really serious effects happening here. And there have been cases where they call it sort of like the boy raised by wolves, where very young children were kind of neglected, and animals I mean, it sounds very far-fetched, but animals kind of help to raise them. They really sort of lose the ability to be able to communicate effectively with other humans. So if this is a short-term thing, recovery is much more possible and higher chance. But if it's a very long-term deprivation, then of course we see some really serious effects happening here. And of course, stress is really important. So stress early in life, we know that the amygdala can increase in size, and the amygdala is important for emotion, especially fear response. We see that the hippocampus is going to decrease in size. Remember, the hippocampus is really important for consolidating long-term memories. And then we also see potentially development of depression and anxiety disorders later on. And what's probably happening here is stress, releases a lot of different hormones, for example, cortisol, and cortisol can have detrimental effects on the brain if the exposure is long-term and can cause also not only disorders like depression and anxiety, but also can cause other physical disorders, physical diseases happening because the body isn't able to we talk about the parasympathetic and sympathetic system, not able to calm the systems down as it would if it was not in this constant 24/7 stress state. So we're going to pick up the next set of slides on abnormal development. We're going to talk a little bit about different kinds of problems that can happen if things go wrong in terms of development. And then we will start from there. So we'll see you next time.